So, hello students. Today, in last lecture, I stopped. Our lecture is stopped at incomplete dominance. Right? Incomplete dominance means we understand here. In incomplete dominance, our phenotype ratio and genotype ratio they are same. Here, phenotype ratio means red. The ratio between red, pink, and white it is 1:2:1, and in genotype ratio it is also that is pure red 1, hybrid red it is 2, and small pure red it is white it is 1, right? Means we understand here phenotype and genotype ratio are same or similar in case of incomplete dominance. In our NCERT, one example is given for snapdragon, that is the snapdragon flower. Other examples are also there. As for example, as far we can see here, in case of first it is given in snapdragon given in our NCERT. Second example in 4 o'clock plant, commonly called as 4 o'clock plant, or another name of this 4 o'clock plant is Guleman, right? Guleman, or scientific name is Mirabilis plant. Mirabilis, right? Here in Mirabilis, or we can say 4 o'clock plant, or it is also called as Gul-E, that is called as Gul-E Barnes, right? Another animals also, this incomplete dominance is also present in animals. As for example, in Andalusian fowls, Andalusian fowl. Andalusian fowl means what? And when we cross between a black and a white fowl, in F1 generation we get blue fowls, right? And in F2 generation what happens? We have that is black 1, these two are blue 2, and white fowl 1. We get the 3 ratios. Means here we understand this incomplete dominance is present in plant and animal both. As for example, in plant snapdragon, right, and Mirabilis, and in case of animal it is present in Andalusian fowls. Okay, now how, now new question is there, how incomplete dominance occur, right? Here it is explanation, it is also given explanation of the concept of dominance, how one is dominant and one is recessive. And here very easy we understand that for dominant means for a phenotypic characters, who are responsible? These enzymes are responsible. That means we can say, okay, enzymes means what? Proteins. They are substrate proteins, right? Proteins, how protein forms? Means we know very well. DNA, it forms messenger RNA. Messenger RNA through translation forms proteins. And these proteins, they are converted into different type of enzymes. And these enzymes are responsible for phenotypic expression, right? Here one thing is there, means enzyme if formed, so it shows its phenotypic expression. Enzyme is not formed, means no expression is there. Suppose that some means inactive genes are there. Inactive genes means they cannot form the enzymes. No enzyme is there, that means their characteristic is not expressed. That is also given here. What exactly is dominance? Why are some alleles dominant and some recessive? That is the question. To tackle these questions, we must understand what a gene does. What is the function of gene? So the function of gene means what? A gene is present. We understand. Okay, gene is the part of segment, right? And it gives code into the messenger RNA. Messenger RNA, it has genetic codes, right? So genes means what? It is responsible for the formation of enzyme, or we can say it is the responsible for formation of protein. Protein means what? We can say enzyme. And enzyme is responsible for phenotypic expression. That is given here. Every gene, as you know by now, contains the information to express a particular trait. Expression means, gene expression means translation. So through translation, they produce proteins, right? In a diploid organism, because here we are talking about the angiosperms, and angiosperms of, we know, pteridophytes, gymnosperm or angiosperm, they are sporophytic, means their dominant phase is sporophytic. Our angiosperm means all the plants are sporophytic. Sporophytic means they are all diploid, 2n. There are 2 copies of each gene. That means as a pair of alleles. So these genes are present as a pair of alleles. Now these two alleles need not always be identical. They are not identical because in sexual reproduction what happens? There are some differences there, right? As in heterozygote. So zygote means capital T, small t is present. That means two different type of genes are there, right? One of them may be different due to some changes that it has undergone, about which you will read further on. Further on means what? Some changes due to the mutation of, due to the crossing over or other phenomena is there, right? We will read in the next chapter, right? This one is in the next chapter in molecular basis of genetic inheritance, which modifies the information that a particular allele contains. Which modified the information. Which modified, how information is modified? Information means what? That is the sequence of genetic nitrogen base, or we can say sequence of nucleotides. So sequence of nucleotide, it can be changed through crossing over or through mutation, maybe through recombination also, right? Which we read in next chapter. Okay, let us take an example of gene that contains the information of producing an enzyme. Now here we are going to take an example, right, in which a gene is responsible for the formation of a protein, and this protein is responsible for the formation of a enzyme, right? Let us take an example of a gene that contains the information of producing an enzyme. Now there are two copies of these genes. Because two copies means what? Why two copies? Because here homologous chromosomes are there. Pair chromosomes are there. One gene is present and another, one pair, another gene is present in another chromosome. So here one pair, right? Simply is given. There are two copies of these genes. The two allelic form in the form of allele. Let us assume, as in more common, that the normal allele produces normal enzyme. Produces the normal enzyme because it is normal, right? Simply what is that is needed for the transformation of a substrate S, right? Here what is given? A normal allele produces a normal enzyme which is necessary for the transformation of a substrate S. Substrate means what? Product, right? Theoretically, the modified allele could be responsible for the production. The normal allele means it produces the normal enzyme, but a modified allele, means in which changing occurs, in which mutation, in which recombination, in which crossing over occurs. Then what happens? It is responsible for the production of, we understand here we have three options. First, three probability or possibility is there. First, the normal or less efficient enzyme. If it is a normal, so it produces normal enzyme, right? Some modification means less efficient enzyme. First thing. Second, a non-functional enzyme. It produces an enzyme, but this enzyme is totally non-functional with no function. It shows and no expression, no function is there, right? No enzyme at all, or this gene is also a silent gene. Silent gene means it cannot express itself. Not express means it cannot produce any enzyme. Three possibilities are there. Normal or less efficient enzyme. Here normal enzyme is formed, means normal phenotype is formed. In dominant case, what happens? This normal enzyme is formed. Normal enzyme, it produces the normal character, phenotype, right? As for height is there, right? So capital T is dominant. That is what? Capital T, it is a normal enzyme, a normal gene, and it produces messenger RNA, and which forms through translation a protein, and this protein is converted into normal enzyme, and this enzyme is responsible for targets. We can say it may be auxin or other things, hormones, right? Second thing is there, a non-functional enzyme. The non-functional enzyme or no enzyme is formed through the recessive traits. Recessive traits, they also forms enzyme, but this enzyme is non-functional. Non-functional means it has no function, no activate enzyme, right? It cannot produce any phenotypic, any phenotype, right? And no enzyme, and if the recessive gene cannot produce any enzyme, right, then means it may be a silent gene. Then what happens? No enzyme is produced, no enzyme is no phenotype is occurs, right? Means phenotype means what? No expression occur, no external feature we can see here. So we understand here only first option is for dominant means dominant genes, they form the normal enzymes, and recessive genes, that second and third, non-functional, either they form non-functional enzyme, because this is non-functional enzyme, so they cannot produce any expression, or they have no enzyme. No enzyme is also they have no function, and that is given here. In the first case, the modified first case means what? It is, we know first case means normal or less efficient enzyme. The first case, the modified allele is equivalent to unmodified allele. That means it will produce the same phenotype or trait. Modified allele, modified means one capital T and small t is there, so that is called modified allele, right? So here it is equivalent to unmodified allele. Equivalent to unmodified means what? Like capital T or small t, it is equivalent, right? So equivalent to capital T is actually unmodified allele. So it will produce the same phenotype or trait, same expression it will produce, and that means result in the transformation of substrate S. Substrate S means the protein is formed. This protein is converted to enzyme, and this enzyme is responsible for the expression of gene or for the formation of phenotypic character, right? Such equivalent allele pairs are very common, but if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. That means that time no phenotype is there. The phenotype trait will only be dependent on the functioning of unmodified allele. Look, the phenotype trait, we can say it is a phenotype or it is a characteristics or property, will only be dependent on the functioning of the unmodified allele. The unmodified allele, that is functioning allele, right, which represents the original phenotype, that is capital T, is the dominant allele, and the modified allele is generally the recessive allele. Hence, modified means which cannot produce any enzyme, or which if it produces enzymes, that enzyme is non-functional, right? So that one is called recessive. Hence, the example above, the recessive trait is seen due to the non-functional enzyme or because no enzyme is formed. Do you understand here? Non-functional enzyme, no enzyme. These are responsible for recessive traits. And functional enzyme, we can say normal enzyme is formed to normal allele, that is responsible for dominant traits, right? So next point is here codominance. Co means both. Both shows their dominant, means both genes they can express themselves separately, right? They are not intermingled. Here also we understand. And incomplete dominance we get when we cross between red and white flower, then we get pink flower, which is not matching with anyone. But in case of codominance, means both allele they can express their phenotype independently, right? Here, till now we were discussing crosses where F1 resembles either of the two parents, that is dominance, or was in between, that is called incomplete dominance. But in the case of codominance, the F1 generation resembles both parents. It resembles both parents, means both characters are present in the F1 generation. A good example is a different type of red blood cells that determined ABO blood group. Here given an example of blood grouping in humans. In blood group, we know 4 types of blood groups are there. That is A, B, AB and O, right? In A, B, AB and O, here codominance is only for AB, not for A or not for B or not for O. It is only for AB, right? Here you understand. A good example is different type of red blood cells that determined ABO blood grouping in human beings. One more thing I tell you here, because here how many alleles are there? Here 3 alleles are there. A, B and recessive, that is small n, right? Because here more than 2 alleles are there, so it is also called as, we can, because more allele we can say polyallelic, right? Okay, here A good example of different type of red blood cells, that is my ABO blood grouping in human beings. ABO blood group are controlled by the gene I, right? Here the plasma membrane of red blood cell has a sugar polymer. Sugar polymer. Which polymer is there? This one is here. Sugar polymer. Where plasma membrane of red blood cells. Sugar polymer. Which one is there? It is agglutinogens, which is called antigens, and they are glycoproteins. Sugar polymer. Sugar polymer means we know polysaccharides. Specific type of polysaccharides are there, right? Sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene I has 3 alleles, polyallelic inheritance, right? IA, IB, and recessive I. Okay, these AB are dominant on I. The allele IA, IB produce a slightly different form of sugar, while allele I does not produce any sugar. You understand here? Because allele small I does not produce any sugar, means does not produce any phenotype. That means what? It cannot express itself. That's why it is recessive, right? But they produce slightly different type of sugars. Because they produce sugars, means they are dominant. Okay? Because humans are diploid organisms, each person possesses any 2 of the 3 I gene alleles. IA, IB are completely dominant over I, right? In other words, when IA, I are present, only IA expressed, because I does not produce any sugar. That is the example of dominancy, right? Who is dominant? That IA is dominant over small I, because small I cannot produce any sugar. Second, when IB and IA are present, then IB expresses, because I cannot produce any sugar, right? But when IA and IB both are present, so both can present together, so they both express their own types of sugar, because both types of sugars are present. In case of IA and IB, both type of sugar is present, both sugar. So here we know antigen, which type of antigen is present? Here both antigens are present. Antigen means A and B, both antigens are present. Both antigens are present, right? That's why it is called AB blood, right? This is because of codominance, and that one is called codominance. I think you understand codominance means what, right? When both express separately, right? Hence, red blood cells have both A and B types of sugars. Since there are 3 different alleles, we know here 3 different alleles are there. So there are 6 different combinations of these 3 alleles that are possible. 6 different combinations, because we have 3 alleles. Here which formula we use? N in bracket, n plus 1 upon 2, right? So means here number of allele, when we read by this formula, we can get how many combinations are possible, right? And therefore a total of 6 different genotypes of the human ABO blood types are possible. How many of phenotypes are possible? So phenotypes means how many? Phenotypes are 4, and genotype means how many? 6. 6 genotype and 4 phenotypes are possible. Okay, okay, we will see this one through. You can see here what is given here. Because 3 alleles are there. Which alleles? IA, IB, and small I, right? Here I represent with, okay, we will do it. IA, IB, and I. What is given? 3 alleles. Because 3 alleles are there, so how many genotypes are there? How many genotypes? Here we understand here, because of 3 alleles, a formula is given here, n plus 1 upon 2. How many alleles? 3 alleles. That means what? 3 in bracket 3 plus 1 upon 2. And when we expand, we understand 9 plus 3 upon 2, and that means 6. 6 combinations are there. That is 6. Right? In case of rabbit, 4 alleles are there. Now, can you tell me how many combinations are formed here? Coat colors of rabbit, it is means due to the 4 alleles, right? So, because if 4 alleles are there, suppose that 4 allele is given, how many combinations are there? Simply we have to use this formula, and you understand n plus 1 upon 2 means what? 4 plus 1 upon 2. That means what? 16 plus 4 upon 2, or I can write here 20 upon 2 means how many combinations we get? 10 combinations. In case of rabbit, I think okay, I expect you understand number of alleles. When we get the number of alleles, we understand how many combinations are possible. Do you understand how we calculate these combinations? Okay. Now here, here it is represented by IA. We have 3 alleles, I, B and small I. And I represent this A through A, B through B and I through O. O means 0, means no sugar. This one means no sugar. This one means B sugar. Right? And this one means A sugar. So both are dominant upon O blood group. Right? Now here we understand. Suppose that a question is given here. A person with A, B blood group when crossed with O blood group. Suppose that O blood group is given. O blood group means what? Only I is there. I write here O. Right? Do you understand? What is the inheritance? It is the inheritance of blood grouping. Do you understand here? That is BO, AO and AO. Means here we understand all these are heterozygous, right? Heterozygous means what? This one, it contains I, B and small I. It contains also I, B and small I, right? It contains I, A and small I. It also contains I, A and small I. Simply no one is AB blood group, only either A or B. Now we have only two options, either blood group A or it is blood group B. But not, this one may be, but not AB absent, O blood group is also absent. When we see the combination, simply when we see the combination, here I represent AB, right? A into cross with B, right? We can write, I can write here. So B, it is AB blood group, AO, that means AO blood group. Now BO, that is BO blood group, right? O, only O blood group, right? Another one is A, that means only A blood group, and B means B blood group, right? How many combinations are there? 6 combinations are there. 1, 2, 3, 4, 5, 6. When we see, look here, that A means what? It represents the IA. So can you tell me what is the, this is the I gene, A, and this is the another I, B. This one is, IA and small I. This one is capital IB and small I. This one is small I, both are recessive. This one is A, this one is B, simply. So these are the 6 combinations. 6 combination of blood group. That are genetic and phenotype. This is the genotype. How many genotypes are there? 6. And when we consider the phenotype means only dominant character expressed. So here we understand that is the blood group A. This one is blood group A, B. This one is blood group A. This one is blood group B. This one is blood group O. This one is blood group A, and this one is also B blood. This side I write here blood group. This one is genes, right? Genetic. So you understand here blood group AB, A, and B, and B, and this one is O. How many? 1, 2, 3, 4. A, B already. So 4 types of phenotype. 4 phenotype. 4 phenotype: A, B, O, and E. Do you understand this one? Right? Which one is example of codominance? That one is example of codominance. That is the example of codominance. So one example of codominance is present in our blood grouping, and that's which blood group? Only AB. It shows the codominance. This one, these two, they shows the dominancy, not a codominant, but dominance and recessive, law of dominance, right? Okay. One thing is there. Is there any other example is also there? Yes, we can see in case of some roan, when we use black and cross with a white cow, right? In F1 generation we get roan color. Roan color means what? Here some black hairs are also there, some white hairs are also there. Both are present. Sometime what happens in dogs, you can see different patches. In cow also patches are also there, right? Skin color, we can say skin color, right? So roan skin color is the F1 generation, and in F2 generation we can. Roan means what? It is the example of codominance. Means the gene which is responsible for black color is also present, simply black hairs, and genes which is responsible for white color hairs, it is also produced. So roan type of roan color of cows we can get. Second, some patches are there. Patches means where white color is there, it is due to the dominance of white color, and some dark is there, it is due to the dominance of dark color, right? So roan color means we can say this is also means when we cross between black and white cow, then what happens in F1 generation, we get all the cows, they are in roan color, right? And that is the example of codominance, right? So codominance. Another one is incomplete dominance. Do you understand what is the difference between two? Right? We understand that in case of incomplete dominance, what happens? Intermediate form is formed. But in case of codominance, both phenotype is present in F1 generation. Okay. Some more other things are also. One thing also I tell you, otherwise you get confused. Here you can get, suppose that sometime what happens, some genes are very fertile. If they are present in homozygous condition, they are fertile. Fertile means they kills the organism in their embryonic stage also. And these type of genes are called lethal genes. Lethal gene. Now question, what is lethal gene? Lethal genes means what? Lethal genes which cause death of individual. Here we can say they causes death of organism or individual right? When they comes in homozygous condition. They are called as lethal gene. As for example, Y, yellow color. In mice, right? This Y, this one is lethal. But when it is present in homozygous condition, right? Okay, this is for yellow color. Yellow color means in nature you cannot find out the yellow color mice, but right, only white color we can get, white and brown color. So why not get yellow color? Because yellow color is obtained only in their homozygous condition, right? So when we crossed between a we can say brown and white color rat, we can express here. We have two types of gametes. Another one is small y, that is brown color, right? That is white color. Then what happens? We have two types of gametes, capital Y this side, right? This side I write small I, right? Because in F1 generation, all these are, this is the F1 generation. Okay, we can write, I use this one. If we cross between capital Y into small y, in F generation we know we get capital Y and small y, right? This is the F1 generation. And when this look at, because it is animal, so here we cannot write it self pollination, we should write here submating. After submitting, what happens here? Submitting, what happens in F2 generation? Here we get, okay, you can see here we get four different type. What different type you can get here? Suppose that I draw here. We have two types of genes here. One is capital Y, another one is small y. Capital Y, small y. So first means capital Y. Here capital Y, small y. This side also capital Y, small y, but this side small y, small y. When it is present in this one, look, homozygous condition. So this is fatal, this is lethal. So when it is present in a single organism, so what happens here? It is responsible for the death of embryo in its fetal stage. Here we only get 2:1 ratio. This 2 and small y ratio we get. Why we get only 2:1 ratio? Due to the presence of lethal gene, and that one is called lethal gene because it is responsible for the death of mice, and it is only active in homozygous condition. So do you understand what is lethal gene? Lethal gene is responsible for the death of organisms when they come in homozygous condition. They may be recessive and they may be dominant, that is depend upon their, right? So they may, mostly they are recessive, right? So it is an example of in animal case. In case of plant also lethal gene is present in snapdragon. This is in rat, right? As for example in yellow color in case of rat, it is lethal as for example, right? And another example is snapdragon. What happens here? Golden color, you can say golden color of leaf is lethal. So these are two examples of lethal genes, right? After lethal gene means pleiotropic gene is also there. Pleiotropic genes, as far as pleiotropic means, it is also given in our NCERT, so we need not to discuss here. We will read the NCERT and then find out the, right? So come back to the NCERT. So I think you understand. Here a column is given. Allylic parent from allylic form parent first, allylic form parent second. IA, then we get IA, that is blood group, right? This one is genotype and that one is our phenotype, right? IA, IB, IB, IB. Here you can see that is our genotype and that is our phenotype. So blood group A is there because both are AA is present. Here AB present, blood group is AB, and that is the example of codominance, right? This one is IA, IB, only A, and that is for dominant character. It's law of dominance. AB, codominance, right? B, dominance. You can see here this B only. This is the pure homozygous, right? This one is heterozygous, only B. This one is heterozygous, and it is for dominant. It shows the dominant. O means it is homozygous, right? We can understand here. This one is homozygous. This one is homozygous. This one is heterozygous, AB, and codominant. Heterozygous. This one is also codominance. This one and this one is codominance, right? That are shows the codomination because both produce their polysaccharides. Okay, now come to the. Do you realize that the example of ABO blood grouping is also provide another a good example of multiple alleles? That is, yes, here because more than multiple alleles. Why multiple alleles? Because more than 2 alleles are found here. More than 2 alleles. That is called multiple allele. Right? And that is more than 2 means how many alleles are there? 3 alleles are present. 3 alleles. That is A, B, O blood group. Capital A, capital B, and I. Right? Allele? Yes. Here we can say yes. Here you can see that there are more than 2, that is 3 alleles governing the same character. Since the an individual only 2 alleles can be present, multiple allele can be found only when population studies are made. What do you mean by population? Look here, individual, and when more than one individual is there, then it is called population, right? So population, when population studies are there, then we understand the multiple alleles. For multiple allele. So multiple allele can be found only when population studies, right, are made. Occasionally, a single gene product may produce more than one effect. Sometimes it happens. A single gene product may produce more than one effect. If a single gene produces more than one effect, then this type of gene is called pleiotropic gene, right? For example, starch synthesis in pea seed is controlled by one gene. That one gene is B. That gene is B, capital. It has 2 alleles, capital B and small b. Starch is synthesized effectively by capital B means large size starch, right? Homozygote, and therefore large starch grains are produced. In contrast to small b which is homozygous, it has a lesser efficiency, so it is the smaller starch are formed. Starch grains are formed, right? Have lesser efficiency in starch synthesis and produce smaller starch grains. After maturation of the seed, when a seed is matured, capital B seeds are round because large sized starch are formed, and small b are wrinkled because more amount of water is there, and here smaller size starch grains are there, right? So it becomes wrinkled, right? Heterozygote produce round seeds. Heterozygote, they produce round seed. It is not intermediate, right? Round seeds, right? And it seems, so capital B seems to dominant element. When we consider the seed shape, then we can say capital B is the dominant over small b, right? But when we consider the starch grain that is given, but the starch grain produces of intermediate size in capital B small b. Here, intermediate size grains are formed, capital B small b, right? So, if starch grain size is considered as phenotype, then from this angle, the allele show incomplete dominance. Do you understand? When we consider the shape of seed, then it shows law of dominance. But when we consider the starch grain, when we consider the phenotype as a starch grain, right? Starch grain size. So large starch grain means where capital, in case of capital B small b, in heterozygous condition, intermediate size is formed. Because intermediate shape and size is formed, which is not tally with capital B or small b, that means we can say it is incomplete dominance. Do you understand? If we consider starch grain size, that is the example of incomplete dominance, and if we consider the shape of seeds, right, that is a law of dominance. It prove the law of dominance. Therefore, dominance is not a autonomous feature of a gene. Dominance is not a autonomous feature of a gene or the product that it has information for, right? It depends as much on the gene product. Is totally depend upon the gene product and the production of particular phenotype from this product as it does on the particular phenotype that we choose to examine, right? In case of more than one phenotype is influenced by the same gene. In case when more than one phenotype is influenced by the same gene. Here you understand, right? What are the more than two effect means what? Here two effects means that seed size, first seed shape and size. Second effect on starch grain, right? So means when we consider these both, we have two different idea. We get first law of dominance when we consider the seed shape, and second incomplete dominance when we consider the starch grain size, right? And the gene which is responsible for more than one phenotype, that type of genes is called pleiotropic genes. Pleiotropic genes. Okay.